Hi, I might as well get started. I'm Bruce Luger, and we have a lab right across the street over here in Building 10 where we do brain stimulation. I'd like to talk a little about that today. I know it's kind of an MRI course, but we do quite a bit of MRI as well, so it works in. So we're in the experimental therapeutics and pathophysiology branch. I'm new to that. I've been here since December. In our lab, we're in the non-invasive neuroscience unit. I was at Columbia before that at first, and then at Duke University, and from there came here. And I've been doing TMS, mostly transcranial magnetic stimulation, for about 21 years now. Forward. Okay, so TMS is part of a more general topic of a non-invasive brain stimulation, or NIBS. I don't like saying NIBS yet, it's still new and sounds silly. But it's basically applying energy to the brain to modulate its function. And you can do that with one new thing we will be trying soon is ultrasound, for instance, targeted ultrasound. You can use light. What we focus on here is using electromagnetic energy and specifically magnetic fields to non-invasively affect how the brain is functioning. What you see here, you see that that's Oscar. He's using a figure-8 coil on a subject. You can see it just place it on the scalp. And what you do is you have a very, very, very fast pulse. This is happening in less than a millisecond, a very, very intense, powerful pulse. The pulses can get up to 2 Tesla, so they're pretty big. They only last, like I said, less than a millisecond. And so this very fast changing magnetic field induces a current in conducting volume near the coil. And those currents that get induced then affect neuronal tissue, for instance, creating action potentials. And I got very excited about this. I started out in MEG. I got my PhD in MEG, magnetic encephalography, and went on to EEG and PET, and finally MRI. But about around 1995 or so, I got a chance to start working with TMS. And I'm an experimental psychologist, so that was very exciting because all those areas I was just talking about are correlational, right? You just kind of have somebody do a behavior and see what happens in the brain and hope that there's kind of a brain-behavior correspondence. But here you can actually do real experiments. Okay, you can act on the brain and see what happens. That's a true causal experiment. So this is like to an experimental psychologist, it's a gold mine to be able to do something like that. So how does TMS affect? What kind of effects does it cause in the brain? Well, first of all, they're acute effects. In other words, effects that happen right away and wear off very quickly too. So, for instance, and it's kind of the way we show that TMS is working and convince you it's actually doing something is we'll put the coil over motor cortex, right, and aim for a muscle, okay, such as the FDI muscle right here in your hand, and we can make your finger twitch, right? And people always find that very amusing, but it's also showing that we're actually affecting the brain by doing that. And that kind of thing is something you can do anywhere in the brain, but you can only see direct evidence of that in certain places in the brain. For instance, motor cortex. You can put the coil back here over visual areas, stimulate, and the subject will see phosphenes, for instance. That's all kind of fun and interesting and everything. You can put it over speech areas and have somebody like I would count down from 10 to 1, and all of a sudden you can't say anything because you've arrested speech because you've disrupted the area involved with that processing. But you can actually do very interesting experiments. For instance, the very first experiment done. Does this have a no? Oh yeah. Oh, good. Okay, so one of the very first experiments ever done was to take the coil and put it over visual cortex. This was done by Vahi Amassian et al. In 1989. And if you just show some letters on a screen, 3 letters, and ask people, what the 3 letters were, they'll be able to tell you. But if I stimulate at a certain time, okay, back here, suddenly they can't tell you, and that's what this is showing here. If I stimulate when I put the letters up, and then there's no time between when I do the pulse. This is the results of 3 different people. Here's how many letters they get right of the 3. So they're able to answer, if I do a pulse right when I show the letters, no problem, they can answer the letter, they can answer how many letters there are. But if I stimulate at around 80 to 100 milliseconds after, which is about the time it takes pattern information to get back here, suddenly they can't answer. Okay, they started calling this a virtual lesion. It's not really exactly that. You're essentially adding a whole lot of random neural noise, right, to an area and disrupting the processing. And by the way, you don't have to just disrupt processing. It's all in when you do the stimulation. So you can actually also enhance processing that's going on and make people better at things as well. And that's something that I've specialized in myself. So you can use these acute effects to just explore what's happening in various areas, in various cortical areas. We've also found out that if you do repeated TMS, say over a number of sessions, you can create cumulative effects. You can create long-term effects that last. First of all, you can do TMS, trains of TMS, for maybe 15 or 20 minutes, and its effects can last another 20 minutes to an hour. Okay, but you can also do that repeatedly, over days and weeks, and create effects that last for months. Okay, changes. And so, for instance, TMS is now approved for depression by the government, so that is actually now you can use it for people that drugs don't really work on, and it's pretty much the only thing that will work for them in depression. And this is just a graph down here over 2, 4, and 6 weeks of the effects active versus sham TMS, showing remission rates and response rates, which are growing over the weeks. Is there? Okay, there's a clock right there. Good. All right, so I think I'm not going to be able to. This is basically 45 minutes to an hour, and I'm not going to be able to do justice to. Over the last few decades, we found a zillion different ways to use TMS now with the brain, because brains are very complicated places, and there are all kinds of things you can do. So I think maybe, though, kind of just as a kind of a very brief overview of it all, I'll do it in terms of this very, very large parameter space that you can have with TMS. So, for instance, just temporally, if you do the TMS pulse by itself, which is most devices create a cosine wave, okay, and again, within about a half of a millisecond. And so, nonetheless, you can actually shape. Now, there's devices that can create arbitrary shapes, like a square wave, for instance. So the actual waveform and its duration can be changed, and so you have an infinite number of possibilities with just the wave form itself. And then, if you do trains of TMS, you've got a frequency involved. And it turns out the brain is very sensitive to the frequency of the TMS. And I'll talk a little about that. And if you do trains, how long is the train? That matters too. How much time is there? That's what the ITI, intertrain interval, is. The time between the trains matters as well. How many pulses you use, how many trains you use, how many sessions of trains that you use, all create effects. And you can actually make things even more complicated. For instance, you can nest frequencies within frequencies, okay, so that you can stimulate at, say, 50 hertz, and I don't know how many of you are familiar with endogenous brain oscillations, but 50 hertz is in the gamma range, for instance, very important functional region of frequencies in the brain. So if I stimulate a gamma frequency, and I just do a small train of that, and then wait 200 milliseconds and do another one, so 5 times a second, I'm giving gamma, but 5 times a second is pretty much in the theta range, which is another functional frequency that we know from EEG. And so I can nest frequency within frequency. That's called a theta burst stimulation, and that is a very potent kind of stimulation and creates other kinds of effects. And then you have your spatial characteristics. You have the coil shape. You have the coil itself, a figure-8 coil, which you see right there, is the most focal kind of coil that we use. Basically, the current is essentially strongest right at the center of the coil, going in a certain direction. It adds up in the middle, and it's kind of less on both wings of it. So you have, but a figure 8 coil has 6 degrees of freedom to it. It has X, Y, Z in space where the center of the coil is, and it can rotate in 3 different ways. Okay, so coil orientation turns out to be pretty important, and obviously its location in space is very important. So you can see just from this one slide that you have a huge set of parameters, and each parameter has an infinite number of possibilities, okay? And we've only just begun to explore this huge parameter space and all the effects that it creates in the brain. So just starting out with spatial parameters, okay, I can talk about target localization. Right? So a TMS pulse can be guided so that it can have a resolution of about a half a centimeter. Okay, you can affect larger areas as well, but you can't really get much smaller than that in terms of the effects that you're going to see. But that's actually pretty good. It's on par really with, for instance, EEG spatial resolution and MEG and even well, fMRI and MRI is a little bit better than that. But nonetheless, it's actually pretty complementary to looking at systems in the brain and looking at individual cortical areas. Individual functional cortical areas are on the area of about maybe 3 square centimeters or so. So, you know, you have a tool that can affect a given, a cortical area. So, well, how do we aim a TMS coil? In the beginning, we didn't really have very good ways of aiming because we weren't tied to imaging yet. In the 90s, the early 90s, things weren't as well developed in the imaging world as they are now. We started out just first by looking around on the scalp itself. So we did experiments like in the visual areas. You could do that, like the one you just saw with Amasian. We replicated that ourselves, and it does work. You can go to the motor cortex. So there's 3 labs here in the NINDS of Mark Hallett, Eric Wasserman, Leo Cohen. These guys basically started a lot of this. This is a great tool for neurologists, by the way, because it does affect corticospinal activity, and you can do a lot of different kind of diagnoses and things with it. And those guys were leaders in the field, have been forever. That's easy to do because you can create a response and you can measure it with electromyography, so EMG, very nicely and with nice quantitation. So that area, half the papers you'll see if you ever look at papers of TMS in the literature, half of them are on motor cortex, just because you have this nice way of quantifying the effects in the cortex, really corticospinal effects. But nonetheless, if you go outside of primary areas, primary motor, primary visual, you can't really see directly what the effects are on cortex. And so the best that we would do then is use the 10-20 system on the scalp, just so at least we would be pretty consistent among subjects about where we were putting the coil. But we have an idea, I'll show in a second, of where over the brain you are, but it's still pretty low resolution. You can go on to use structural anatomic MRIs, and you can go beyond that by using functional fMRI with group analyses to kind of get to avert a certain spot where on average there was a functional effect. Or you can, and the state of the art now is to do this on an individual basis. I can put up somebody in the MRI machine, get fMRI on them while they're doing a task, and then I can aim my coil right at that spot that got activated. With just scalp localization, this is just an example. You can see it's from 1987 of just those shaded-in regions. These shaded-in regions here are areas where you can't quite see them. You can see the little dots here, but that's where individual subjects end up, where the electrode wound up being when they did some CAT scans over them just to see where they were over cortex. And you can see that oops, I lost my, there it is. The resolution isn't great. You could be anywhere in this general region if you're using this particular electrode site. So again, not very good spatial resolution. That in the 90s, that was pretty much the best we had. What happened was we developed ways you can use infrared technology to, in space, know exactly where somebody's head is, co-register it with their structural and functional MRIs, and know exactly where the coil is, all with millimeter resolution. So I can actually now aim, say I just want to aim at a given spot in the brain, I can do that on an individual with millimeter accuracy with a frameless stereotaxy. So it's a very nice tool, and it's really hard to function without it at this point. Just for an example, just to look across these various ways of targeting, a group led by SAC in 2009 published, basically and as I show these different parameters and stuff of TMS, I'll also try to show you some of the various paradigms that we use to do experiments with TMS. But so for instance, you can do a Stroop-like task, visual task, where I can say, answer which is the bigger number. And I just mean the cardinality, the larger number, like 4 is larger than 2. So you'd answer, when you see this 2 and a 4, you would answer 2. But what happens if I make the 2 bigger? That actually makes it harder to do the tasks. It slows you down, okay? And if I make the 4 bigger, that actually speeds you up, okay? Because it's bigger in 2 different ways. And so that's kind of a Stroop-like effect. And it turns out that when you do that, and that's what I'm showing here, your reaction time, if I show you the incongruent pair, you're slower. That's what this bar is on average, versus the neutral and versus the congruent, where you're much faster. Okay, so this is what happens in a normal case. And so if I just do sham or pretend TMS, which is, by the way, very difficult to do because TMS makes a sound and it feels like something. And how do you do that without affecting the brain? Well, that's not so easy, but there's ways we can do that. Or approximate that. But at any rate, when you do sham, you just see what happens without TMS, essentially. If I stimulate the right parietal area, I change things where I actually kind of reduce this effect, okay, and actually make it bigger, okay, if I stimulate left parietal area. The point is I can make a significant change in the reaction time that people have in this task, and I can actually do it in opposite ways in different areas. But if I just stick with the right area, okay, and just do this experiment on people, and then I just target in different ways, the 4 different ways I mentioned, using the scalp electrode site versus using structural MRI versus using group anatomical data versus using individual fMRI to target with. What I find, and so that's those 4 different cases. What I find is when I just use the 1020 system, when I'm going to P4 in this case, where is my right side? That's over here. So if I'm going right about here, all right, but I'm using a scalp location, you can see that there's a lot of variability in terms of where you actually end up, what part of cortex you end up over. That variability has nothing to do with where the functional area is that's involved with this task. You're only approximately in the right area. If I use group MRI, fMRI, or use MRI, anatomical, it's just a structural, I can know where exactly in the brain I'm stimulating or stimulate in a functional area of my choice, and that's better, all right? But if I use an individual MRI, so I get a certain activation area with each person that has to do with the task and stimulate right in the middle of that or at the highest, the peak change, in the contrast, I can, now I'm aiming, you can see there's certainly variability in where I'm stimulating, but now the variability is capturing the functional variability, where it is in that person's brain that they're doing this processing. And so they did a power analysis comparing the 4 methods when looking at alpha level, and so seeing how to get up to a P of a 0.05 level, how many people, and that's what's on the x-axis, how many people would you need to achieve that significant effect size, okay, in the group. And if you used the 1020 system, the EEG scalp system, right, you'd need about 47 people. That's this line right here to get up to a P of 0.05 level. But if you use this individual targeting, you'd only need 5. Okay, that's this one right here. You get there almost immediately because you're targeting in a much more functional way. And I don't do any TMS experiment now without MRI, and this is the way I target, is with fMRI. because it just works so much better. You have a much better chance of stimulating a region, that's specifically involved with the task that you're doing. And a way to actually make this better, and I had this at Duke, I don't have it up here yet, is you can use a robot. So humans are actually putting the coil on, and they're either holding it, or you try to have a coil holder, and you're screwing it all on and everything. And by the time you're done, even though you have this nice neuronavigation system, you're still not exactly on where you should be, just because a human's doing it. And there's, like I said, there's 6 degrees of freedom, so that the coil's orientation may not just be right. And if you try to replace it on the person, say they stop and, between conditions and stuff, you don't always replace it exactly the same way, even with neuronavigation. Humans just aren't that good at a lot of these angles and things. But a robot is, okay? And the robot we had, this particular system, could put the coil back on with millimeter precision in all 6 degrees of freedom and keep it that way. And so subjects also move, okay? They fidget, they fidget and sink in their chair. Sometimes they don't want to be near the coil, things like that. So this robot actually keeps the coil. If they move, the coil moves with them, so they can't get away from it. And within 300 milliseconds. So it's just a really nice, precise way of targeting. So we have beautiful ways now. It's taken about 10 or 15 years. Don't look at that yet. We have beautiful ways of exactly with millimeter resolution, getting and keeping the coil aimed at a certain place. And we do even better than that, because we can model people. We can model the electric field that is created by the TMS pulse. We can, because this is all geometry, this is all the physics that, if you know MRI physics, you know this physics as well. And we're able to look at the anatomy, figure out, how the gyri are lining up. We can precisely place now, if we want to, a certain amount of dosage, of magnetic field, of electric field, at the spot in the brain that we want. Which is wonderful. Now, that said, by the way, if I'm stimulating and creating action potentials, I'm not just stimulating this area. If I'm stimulating here, I'm stimulating a network, whatever it's connected to. So much for spatial resolution. Well, you actually have, you still have precise resolution, but you are stimulating a network, and people have to remember that too. And the nice thing about MRI, the nice thing that we can do is we can do TMS in the MRI machine. So TMS pulse is so short that you can actually fit it, okay, in between the MRI pulses, okay? As long as you stimulate with a TMS pulse and give it at least, say, 150 milliseconds or so before the next MRI pulse, okay, you don't create noise. So you can interleave the TMS pulses between the MRI pulses, and you're able to actually create fMRI and see how the TMS pulse is affecting the brain. So you can actually see the functional network that the TMS pulse kind of illustrates. One just quick example of using that in an experiment. So here's a working memory experiment where I have faces and places. I have some houses, pictures of houses here, pictures of faces here. Two of the places so house stimuli especially light up this parahippocampal area, and face stimuli very much. They light up other areas as well, but they especially light up, the faces especially light up, I've lost my, oh, there it is, the fusiform face area, as well as many of you probably have seen this kind of thing before with MRI. But I can have you remember, say, 3 of them, okay, and then wait a little bit of time and then show a picture of a place and say, was that one of the 3 you just saw, right? And you answer, and that's that. It's a nice Sternberg delayed match to sample working memory task. If I take TMS and stimulate, okay, the dorsolateral prefrontal cortex, that area has a lot to do with dealing with distractors, trying to deal with when things are a little bit more difficult. So if I throw distractors in the delay period, so here's some house distractors in this face task, okay, you actually get a little bit worse. But presumably the dorsolateral prefrontal cortex is helping out these processing areas when there's distractors, okay? And it turns out you can actually show that. So when there aren't any distractors, TMS to the dorsal prefrontal cortex doesn't really have any effect. But when I do some TMS pulses during the delay period, disrupting what's going on in dorsal prefrontal cortex, I suddenly, if I'm doing a face task, I suddenly significantly increase the activation in the fusiform face area. Okay, so there's compensatory activity in a far distant area because of something I'm doing up in the prefrontal cortex. And the opposite happens if I do the same thing for the house. I increase compensatory activity in the parahippocampal gyrus. So I'm actually able to show effects, distant effects, due to the TMS, and show how one area is involved with others in a task, in a causal way. Okay, this isn't correlational. I don't have to do a lot of hand waving and explaining. You know, I'm literally affecting what's going on. And then you can kind of see the interactions of different areas. And so a lot of that's been done now, but using TMS and fMRI together really increases the power of both, you know, how well TMS explores the cortex and how well you can explain what's going, you know, fMRI activations. Another good example of something we've done is one thing about TMS. You can see we've modeled with electric field. Here's the TMS coil seen on its side. The electric field created, basically when you get down to about here, a few centimeters in, that's the kind of the green blue boundary there is where you stop creating action potentials. All right, the effect's getting weaker and weaker with depth. All right, so one problem with TMS is it only goes so deep. All right, so you know it's easy to affect more superficial cortex, but say if you're interested in the hippocampus, or say you're interested in stimulating, in the case of depression, you may be interested in stimulating Brodmann's area 25. That's actually a kind of a big deal area right now in terms of deep brain stimulation, for instance. And I can't get there directly with TMS, okay? I can only get to about here effectively. But I can use DTI, so I can do I can have a first TMS imaging session, fMRI session, and get some DTI data. Right, I can seed the target that I'm interested in and see where the white matter comes out. Okay, then just to show that I can affect it with TMS, I can stimulate wherever it comes out on an individual. Say for area 25, one of the places the white matter tracks come out to is the frontal pole. Okay, and I can stimulate that on a person and see and increase, like do a single pulse, but then do an increasing pulse and see if I'm actually creating increasing activation in area 25. And that's, you know, in a second interleaved TMS fMRI session. And so here's, for instance, if I do this tractography, that's just kind of a group average of the white matter pathways. Here are in 10 different people where they come out on each person, kind of just showing us a certain amount of variability to that. But what happened was when we stimulate with larger and larger intensity of TMS, we created more activation in area 25. All right, this is just an ROI of just area 25, and we just increased with increasing intensity with TMS. So I can actually aim anywhere in the brain with TMS if I have imaging and if I can do the DTI. So you know, TMS isn't quite as limited as some might, you know, assume. Okay, so other aspects, spatial aspects of TMS are, for instance, the coil shape. So this is pretty much the main thing that we use with TMS. It's highly focused. The magnetic field is just strongest right in here. The current is going in this direction. Well, it depends, but usually it's going in this direction, you know, kind of with the handle. You can also use something called a double cone coil, kind of fits over the head. That goes a little bit deeper. But it also affects a larger area, and that's again something we show down here. But one of the first things people used was a round coil, but that's not very focal. It just makes a toroid right around this area. There's other kinds of coils you can have. One that was developed or partly developed here in Mark Hallett's lab is the H coil, which is essentially a very large round coil, and it can stimulate a very large amount of area. And in fact, that's what we're showing down here. So here's Zi Deng, who's just started. He's in our lab now. He was with us at Duke as well, and He's come up here now, and he's starting this week, I hope. But what he's done is modeled all these different kinds of coils that people use, and by the way, including MRI coils, because they actually seem to affect people's brains as well. They're not just something that measures. There may be some indications that even the magnetic fields of the MRI coils are affecting people. What you're measuring is sort of a moving target as well. These are all different sorts of coils that Z measured, or modeled rather. Here is the general depth that they reach from the surface of the cortex, and here is how much they're spread. So what's happening is you can achieve greater depth, okay, but at the price of greater spread, greater spatial, lesser spatial resolution. The actual shape of the coil matters quite a bit as well. Also, the orientation, for instance, with a figure 8 coil, how you orient it relative to gyri, for instance, matters quite a bit. And there's reasons for that have to do with how you have different ways, or rather different densities of fluids versus gray matter versus white matter, and directions they are all moving in. The way the freedom the way that the currents can move in the brain creates. What happens is if you stimulate with the current going across, say, motor cortex, that actually creates more activation than when you went with the gyrus. So even the orientation of the coil matters. So it really matters to do get a lot of imaging. And do realistic head modeling if you can, if you really want to know exactly how you're affecting the brain with TMS. Another thing that I consider a spatial parameter is intensity of the pulse. And so the higher the intensity, the larger the area, and the deeper you go. Okay, that's very clear. And by the way, with that, and that's what this table is down here, and actually this table had a lot to do with, was mostly made up here, is the safety table, okay? Really the main risk of TMS is seizure, okay? You can get a seizure from it. It's happened about 26 times now over the last 20 years, okay? They meaning actually the NINDS people especially, for instance, Eric Wasserman, found the safe regions you can use TMS. So the higher the frequency, the bigger chance of seizure. The greater the intensity, bigger chance of seizure. And so we were actually able to create tables. If you stay within, so this is a certain way to measure intensity in terms of motor threshold, which is how much intensity it takes to move this muscle, what the minimum amount of that would be. And that's how we use, that's how we dose the TMS. If you stimulate at, this is increasing intensity, and this is how many seconds you can stimulate with a given train with these different frequencies. So, if I use 25 hertz, and you're stimulating at 100% motor threshold, don't stimulate for a train that's more than 1.3 seconds long, okay? Or else you might cause a seizure. So as long as we stay within those boundaries, it's safe. And in 20 years, I've never created a seizure in anybody. I've done many thousands of sessions at this point. The other interesting thing, though, is we've just barely started exploring intensity, all right? And even subthreshold, even TMS pulses that don't create action potentials can still affect neurons in a number of ways, all right, in very subtle effects that you can see cumulatively. You can actually also see them, for instance, if I do a pair of pulses and I do them a millisecond apart, and the first pulse is so low that it wouldn't actually cause a response in this muscle, but if I put that right before one that would cause a very large response in this muscle, I can actually totally inhibit that second pulse. Okay, so I can actually start looking at interactions between inhibitory and excitatory neurons and neurotransmitters by using paired pulses. There's a whole lot of little games that we play with TMS and in different ways of using it. So anyway, so I've just kind of gone over a lot of spatial parameters. There's also a lot of temporal parameters, which I mentioned in the beginning, such as waveform and frequency and so on. And for instance, waveform, Angel Peterchev, who we've worked with for a number of years now, at Duke, has created devices where you can actually shape the waveform in a certain way. And this is the typical monophasic waveform. You can also do a biphasic waveform, which looks like this. These are the two traditional waveforms that you see with TMS, okay? We can actually shape monophasic and biphasic square waves, okay, or much squarer waves, right? And one typical TMS paradigm is I can inhibit, okay, I can lower cortical excitability if I stimulate at low frequency, for instance, 1 hertz, 1 time a second, over say 10 or 15 minutes. So if I do that, and I do that over motor cortex, I can make, and I look at how big of an EMG response I get from stimulating before and after I do that 10 or 15 minutes of 1 hertz. I'll see changes, and what I'll usually see is an inhibition of the test response, after that train of 1 hertz. That's a very typical paradigm. That's another thing people were calling a virtual lesion. They'll stimulate over an area and say, well, I've reduced its processing power, I've reduced its excitability. That's sometimes true, it's not always true. There's a lot of variability between people, and we're working on how we can capture that and control that. Which I'll mention a little bit later. So we did this typical paradigm where we measured MEPs using a monophasic waveform to motor cortex, and we gave people one of these 4 possible waveforms at 1 hertz for 15 minutes. And when we measured the pulses afterwards again. And compared to this baseline, what we saw was when we did the typical waveform, the typical standard biphasic waveform, we actually didn't create any inhibition. Inhibition is below baseline here, okay? But with these square wave ones, we did create inhibition, and you can see it wears off over time. So this is one of those cases where if you ran 15 people, you wouldn't have seen an effect with a standard TMS setup. And then you go, oh, well, my experiment didn't work. No, maybe if you ran some more people, you would have seen an effect. But if you had a more effective waveform, we see an effect. So we're just starting to explore all the different kinds of waveforms you can use. And by the way, what hasn't happened yet with TMS, and there's reasons for it, mostly due to artifact that TMS creates, is to look, say, at animal models. Okay, so there's been very little work done in animals. There's been some. And at Duke, again, Angel Peterchev has found a way to create pretty artifact-free ways of measuring single cell electrophysiology directly underneath the coil, right after a stimulation. So we're starting to learn more about, for instance, how these different waveforms affect local activity, but that's just something that's just starting now. So most of what we know is basically the effects that we see on normal subjects, for instance. But we're kind of working backwards from what people usually do, but now the animal work is starting. Frequency also matters. This is kind of an earlier study in 2000, as a matter of fact, a PET study. If you use, this is 20 hertz stimulation to prefrontal cortex, you can see how it stimulates quite a bit of area in the brain. But this is all pre-post activations, okay, pre-post contrast with TMS in the middle. You can see that 20 hertz activates, and upregulates excitability in the cortex quite a bit. 1 hertz does the opposite, and you don't see as much, but you can see some blue here of a downregulation of cortical excitability when you use low frequency. So it's very highly frequency specific how TMS affects the brain. And so, by the way, just very quickly in terms of another working memory experiment, this time done with letters. So here you see a letter. I wait 7 seconds and I show a letter. And I say, is that the same letter as you just saw? Here you'd say yes, as fast as you can. In this trial, you would say no. Okay. This is a typical Sternberg experiment as well, delayed match to sample. And this is just showing over here, as you increase the number of letters, you increase the reaction time. So when we looked, we did some MRI, fMRI with this task. We actually lit up parietal area, and you can't see it here, but dorsal prefrontal. And so we stimulated those two areas, and we stimulated at 1, 5, and 20 hertz, first of all, during that retention, that delay period in the task. When we did, 5 hertz worked. The other two frequencies didn't. And at 5 hertz, active compared to sham, TMS, this is active, this is sham. Hello, this is sham. We actually sped people up, whether we use a set size of 1 or a set size of 6, okay? So here we were making people better at the task, okay? By only at one frequency and not at the two others. And when we repeated the experiment, we again got that effect with, here's with 6 hertz, set size 6 I mean. This is 5 hertz stimulation to this parietal area where we saw this effect. And again, we saw that, this is in 21 people now, and we did not see it when we stimulated during the probe period.
All of these reaction times, did they only include correct
Yes.
responses?
Yeah. Yes, and these are median reaction times too, because they're skewed distributions. So when you stimulate, within the task mattered. What frequency you used and where you stimulated all matter quite a bit. And so you have to do these kind of parametric pilot studies to try to figure out how to, and especially the fMRI part, to figure out where and when and how to stimulate. And so, for instance, the NIA, National Institute on Aging, has given us lots of money, this was at Columbia and Duke, to look at, can we affect memory, all right, in older people, all right, which is, when you hit about 70, your working memory starts going, everybody, okay? And so this is actually a very interesting topic to people that are studying aging, and to me especially as I'm aging. But what happens is when we're using this, you can see this kind of green area. This is an fMRI network that was associated with the working memory tasks that we used, the same working memory tasks as we just showed you. When I stimulate lateral occipital complex, in both old and young people, okay, if I stimulate, again, during that retention interval, I actually make both the old and young people a little faster in the task, okay? And if I stimulate in premotor area, okay, premotor area may have a lot to do with, in fact, there's a pretty good chance that it has a lot to do with rehearsal. So I got these letters that I'm looking at, and I just say them to myself silently, okay? Well, this area has a lot to do with that rehearsal process. When I stimulate during the retention period, I made everybody worse, okay? Because if you stimulate during the processing that's happening, you're disrupting the processing. If you stimulate right before the processing happens, so which means that if I'm stimulating during the delay period, the processing that's important here for the task is in the probe period, in the test period. I'm stimulating right before that. I'm not disrupting anything. I'm exciting the cortex right before it's going to do its job. When I brought people back and stimulated right before the trial started in the task, the young people all got better. They got more accurate and faster. Okay, so it really helped them a lot. The older people, it helped them a little bit, but they still were getting a little bit worse, and we're still working on that. But at any rate, so again, when you stimulate, it makes all the difference in whether you can disrupt things or make people better at things, enhance processing. That also tells you a lot about what and when, the dynamics of the task processing. Another thing about frequency is it seems very much tied to the endogenous oscillations that are happening in the brain. So for instance, there's a mental rotation task, okay, a very famous one that Shepard did in the late 60s, early 70s. Another group did TMS, and they did it at the both frontally and parietally they measured each person's individual peak alpha frequency and stimulated right before they did the task, each trial at that frequency. And when they did, they enhanced people's accuracy at the task. Okay, when they stimulated they took the peak alpha frequency and stimulated at 3 hertz less than that, nothing. They weren't, there was no improvement. And when they stimulated 20 hertz, there was no improvement. Okay, so and I don't have time, but there's more examples down here. But the idea is that if the reason frequency matters may have a lot to do with dynamic oscillations that are going on in the brain, and so it's a great way of studying dynamic activity in the brain. And that's another aspect of all this. We got really good at aiming and knowing exactly what we're affecting spatially in the brain using TMS. Unfortunately, there's a huge source of variability, which is we're kind of blind temporally, all right? When we stimulate, we don't know what the brain is doing at that moment, all right? And it changes all the time. So what you really want to be able to do is measure, say with EEG, for instance, the oscillations that are going on, and stimulate, with those. So there's a lot of work we still have to do trying to integrate our stimulation temporally, okay, with ongoing brain activity. And I think when we do that, we're going to have a very, very powerful tool that'll work in everybody, all right, and will be a much better tool to be able to actually examine what's going on in the brain and to do therapy. There's a second talk, by the way, that I'm not giving in a few weeks. Holly Lisenby will talk about things like how you can use TMS in various paradigms to do this kind of exploration, and also as a way of creating therapeutic things, for instance, in depression and OCD and schizophrenia. I do cognitive enhancement. I actually had an R01 at Duke, which we still do, sort of. From the NIA to create cognitive enhancement in elderly people. Hopefully we can use that in dementia and Alzheimer's. And so if we can get a handle on the temporal aspects of things, I think we're going to have a very, very powerful instrument. Here's another quick example of a frequency effect, namely, and this was actually done again by Eric Wasserman's group here at the NINDS. When you stimulate, remember I mentioned this 1 hertz paradigm, where you stimulate 10 minutes, say, with 1 hertz, right? That normally creates a certain amount of inhibition, and that you see right here. So here's baseline, and then this is it. The amplitude of the test, the probe MEPs, the EMG response to motor stimulation is inhibited. It goes down. The amplitude goes down after 10 minutes, okay, of 1 hertz TMS, and then it recovers back to baseline. If I stimulate first with 6 hertz excitatory stimulation, I actually create a huge inhibition that lasts a long time, okay? So I have an interaction between low and high frequencies. It's actually creating a much bigger effect, all right? The hand-waving explanation as why this works, by the way, is that if you think about, we see this all the time with TMS, is the brain doesn't really want to be changed, and it always tries to go back to whatever state it was in. So homeostasis is very important in the brain, okay? It's always trying to get back to the state it's trying to keep things regulated at. So if I stimulate with high frequencies, I'm actually upregulating cortical activity, but that lasts a certain amount of time, and then it's being fought. The brain's trying to bring it back down to the level it was at. Okay, so it's trying it. So then when I start with this 1 hertz stimulation, which is going to bring things down, the brain's already in a homeostatic process of bringing cortical excitability back down. And now I'm adding to it by something that also brings it down. So I'm kind of creating a synergistic effect and just making it overshoot its target and really go down. Okay, that's the explanation that we've been giving for that particular phenomenon. Another example is the theta bursts I mentioned early on, where you have the gamma nested in theta, a pattern of stimulation. It turns out, and I don't have time to go through this very complicated slide, but this is called ITBS, intermittent theta burst stimulation. If I stimulate for 2 seconds, wait 8 seconds, and stimulate again, and do that for about 4 or 5 minutes, okay, I actually excite motor cortex, and I do it for a very, very long period. It's a very potent effect. If I just do the same thing, but don't put an intertrain interval in it, okay, just stimulate continuously, I actually inhibit. Okay, and that's shown here, and here's over time, but this is baseline, and this is with no theta burst effect. Here's where I do the intermittent, and I create an increased excitability. If I just do it continuously, I create an inhibitory effect down here. Okay, so it's the same frequencies. All I did was put an interstimulus interval in there, and nothing happens if I make the interstimulus interval a different timing. Okay, kind of this is intermediate TBS. So I do 5 seconds and then I wait 10 seconds and do that, then nothing happens. Okay, so intertrain interval seems to matter quite a bit. Again, we're still trying to work out why these are all true. It's got a whole lot to do with endogenous, inherent oscillations that are going on in the brain. How these are all functional. How these different areas of the brain are talking to each other. How it's using this timing of activity between areas. And we're getting involved with that, but we're only kind of half blindly doing it now, and we're learning. We can create amazing effects with by working with these oscillations. Again, I've shown you this slide before, but another very important thing to remember is duration as a parameter. So we can create effects that last 50 milliseconds, acute effects, or a second and a half or 2 seconds. We also create effects that last weeks and months. Okay, there's ranges of time that we're creating effects in the brain depending on what we're actually changing in the brain. And this idea of creating neuroplastic effects, which again Holly Lisenby will talk more about in a few weeks. Here's just one quick example of this is called paired associate stimulation, PAS. So if I stimulate the motor cortex, I can create a MEP in the hand, an EMG wave in the hand. If I do, and he's not doing this, he's just no way to draw that right. But if I stimulate median nerve, okay, and then 23 milliseconds or so later stimulate motor cortex. 23 milliseconds is about the time it takes for this afferent stimulation to get to the brain. So they're both coming to the brain at the same time. So I'm actually doing a fire together, wire together situation. So if I do that with 90, say, pairs of these, I'm creating this where I have 2 different kinds of stimulation impinging on motor cortex at the same time. I enhance, okay, the responsiveness. All right, this is a measure of plasticity. Okay, I can actually look at measure cortical plasticity by doing this particular paradigm. Okay, but it's another way, and Ulf Zeeman and his group was just showing by doing this PAS procedure along with practice of a motor task. This is with the PAS with the correct timing. This is kind of with the PAS with the incorrect timing. And this is a control situation. As people practice, they get a little bit better. This is accuracy in the motor task. But with the PAS procedure, you're getting better at the task faster and at a higher level, and you continue that over the next block of trials as well. Okay, so you're actually able to create these plastic effects, and you're able to show that actually translates to something functional as well. Again, these are all different things we can do with brain stimulation now. And in terms of being able to see what we're doing, not just in terms of spatially, but temporally, and in this case, spatial temporally, you can actually use EEG and measure at the same time as you're doing the TMS. You have to turn off the amplifiers for a millisecond right when the TMS pulse is happening so you don't swamp the amplifiers. And this is one of the very first examples of this, but this is El Magnani's group in Finland. They stimulated motor cortex, all right, and created a certain amount of activation at 3 milliseconds. This is what the activation looked like, 7 milliseconds later, as it goes into premotor areas, all right, from the motor area. 1 synapse away at about 7 milliseconds later, it goes across the corpus callosum. So at 24 milliseconds, you're seeing activity in the other motor area. Okay, so you actually can follow and watch the effects of the TMS pulse dynamically, across the brain with EEG. And here's another example of using it for neuroplastic measurement. You can actually use, I'm not going to explain this whole thing as I own time, but you can actually, the blue here is just a baseline response to the TMS. So you have an EEG response. I do a TMS pulse, and I just measure at a bunch of electrodes, and that's just at one electrode, the response to that TMS in the cortex. You can see that if I do a whole bunch of 5 hertz excitatory stimulation, 1,500 pulses worth, so for over about like 10 minutes, I can then enhance, okay, the responses Okay, and you can measure that with ERPs as well. And we call these TEPs, or TMS evoked potentials. Another way of measuring neuroplastic effects of the stimulation. And again, these things, for instance, we're about to use this in a ketamine study in depression and see if we can look at changes in plasticity, because ketamine also affects plasticity as well as depression, amongst other things. So it's a nice tool to use to measure dynamic changes in the brain over a period of, in this case, hours and days. And by the way, just to be complete, so I've been talking about TMS, all right? So it's magnetic stimulation. There's another thing right now that's gotten very popular. It's called TDCS, okay, which is transcranial direct current stimulation. I didn't believe it would work because basically they're taking 1 or 2 milliamps, okay? They're taking 2 electrodes, they put one here maybe, and most of the current's going to go across the scalp, first of all. This is 2 milliamps. It's hardly anything. And a little bit of it's going to go through the brain. And I said, well, that can't do anything, but I'm wrong, apparently. So again, the Wasserman group here led the way in the early 2000s and showed, for instance, with verbal fluency task, okay, that people could actually, that a certain kind of the TDCS stimulation, you know, when you put this TDCS electrode over one of them over language areas, prefrontal language areas, actually increased verbal fluency. For instance, if you do this for like, it creates an effect after about 15 or 20 minutes. There are questions whether it's actually working. People really want it to work. They really want it to work because like TMS can cause seizures, right? I never have, but it can. But this is totally safe. The most you can do is create like a sunburn-like effect with electrodes if you don't put them on right or don't monitor them. But you're not going to hurt anybody. So they're very, very safe. It's really, really cheap. It's the easiest circuit in the world to build. It's just a constant current device. It's the first thing they ever show you in electronics as a circuit. So it's cheap, easy, totally portable, totally safe, right? So everybody wants it to work. But the problem is everybody's just been slapping it on the head and trying different tasks and saying, oh, it's working, it's working. And it really, so some 3 very large meta-analyses and reviews have come out in the last few years that have just said, look, you know, there's no replicability so far. People are, you know, and it's not necessarily that it doesn't work. The problem is that people aren't doing the good science that it takes to, you know, replicate what's found before, use bigger sample sizes, model, you know, where you're putting electrodes a lot better. Do all the things that we're doing with TMS now, they really haven't done with TDCS yet. So it's a real question. You know, there are a lot of claims right now. And I hope it works because, like I said, it's cheap, easy, and fun to use. And it won't hurt anybody. But the jury's still out on it. So, you know, we'll have to see. But there's every indication that it does work. There's a lot of animal models, for instance, even from the 60s, that says it, you know, there's reasons to think that it should work. But I'm running out of time. That's what I just said was all this stuff. So where we are right now is with TMS, okay, is that we're really good, as I just said before, we're really good at spatial parameters. We're not so good at temporal parameters as yet, so that's a developing area. But what we are actually developing is a very, very, very powerful tool to use to experiment in a causal way brain systems, brain organization at a systems level. And so I'm very, very excited about it. I think we've come a long way in the last 15 or 20 years. I think we have another 15 or 20 years to get there. But when we do, this is going to be a tool that when you use it with brain imaging, you know, we're going to be able to really examine the brain in just, you know, some really nice systematic ways. And that's it. This is our group and some collaborators. Any questions or?
Do you see any validity in combining stimulation techniques?
So we were just asked. I was just asked in terms of combining simulation techniques, for instance, like TDCS and TMS. So for instance, the experiment I showed, one of the experiments I just showed, where they did 6 hertz stimulation and 1 hertz stimulation. They've also done TDCS and then TMS, and shown that the TDCS had a very powerful effect on what exactly happened with the TMS. It actually created opposite effects with it. So yeah, no, there's probably going to be, especially as we learn more about the temporal aspects of things, there's going to be a lot of ways, yes, that we can combine electrical with magnetic stimulation that may actually help and may produce more potent effects. One problem with TDCS also, TMS you can do experimentation on the millisecond level, right? You can do one pulse and you know, things happen over 50 milliseconds. With TDCS, you have to wait for a cumulative effect to happen, so it takes 15, 20 minutes to actually see an effect that, you know, that happens. So all you can do is mostly pre-post experiments, but you certainly can use it to potentiate things, you know, in the beginning. Okay. Well, thank you.